Good evening, and welcome to another Wednesday session of the Faith and Science Fundamentals webinar series. This is our 10th week and the second session of the module dedicated to biology, in which we will talk about genetics. DNA and design. Last week, we talked a little bit about DNA and proteins. We said that DNA is made up of 4 types of pieces called nitrogenous bases, and that the proteins are made up of amino acids. I also showed you an image as a preview of what we were going to see this week. We said that DNA and proteins are independent, that they are like the egg and the chicken, that without DNA, there can be no proteins, and without proteins, there can be no DNA. Well, this is exactly what we're going to explain today, but in detail. Let's start from the beginning. Before attending this webinar series, what would have come to mind when you heard the word proteins? I'm sure that it was something similar to this picture. Food that's rich in proteins. Meat, fish, eggs, legumes, or beans for the vegetarians. Why is it important to include proteins in our diet? Well, they're important because they are the building blocks of our bodies. The raw material to make our cells, tissues, and organs. But proteins aren't just building blocks. It turns out that they're also the tools that place the blocks in the right places in our bodies. And not only that, they're also the machines that are in charge of coordinating the building and making sure that everything that's happening in the body happens at the right time and in the right place. We have proteins everywhere. Hair is made of proteins. Skin is made of proteins. Muscles are made of proteins. And this is at a macro level. But if we go to a cellular level, it turns out that proteins are very abundant and very important molecules. Why? Well, because proteins are used for almost everything. They have a lot of functions. We've already said that they serve as construction materials, but they also have a transport function, moving things from one place to another. Hemoglobin, for example, transports oxygen from the lungs to the rest of the body. Other proteins are the body's mail carriers and are dedicated to carrying messages from one place to another. Do hormones sound familiar to you? If you're an adolescent or have adolescent children, you probably know what I'm talking about. Hormones are messenger proteins that at certain times travel from the brain or endocrine glands to other parts of the body and give them instructions to start growing, producing substances, developing hair. Some proteins work like a customs office, controlling what goes in and comes out of cells. Other proteins have a defensive function, such as antibodies that are responsible for protecting us from infectious agents such as viruses or bacteria. Last but not least, we have enzymes that are special proteins that are responsible for controlling the hundreds of chemical reactions that take place in the body. We're going to look at some of these proteins in action. In this animation that I'm going to put up on the screen, we're going to see 3 different proteins. The first one that we're going to see is a digestive enzyme. It's in charge of breaking down our food molecules into small enough pieces for them to enter the cells. The second here is found in the cellular membrane, and its function is to control what does or does not enter the cell. It's this last one here, which is my favorite, which is called kinsine, or kinesine. And it controls things that go from one place to another inside the cell. So the road that you see here is not a fixed road. It actually is built as needed, and it is made of proteins. Even though these proteins seem very different from each other, they all actually have the same basic structure. They are long chains of amino acids. This part is very important, so to make sure that everyone can understand it, we're going to explain it with another animation. By the way, both this animation and the previous one are excerpts from an Illustra Media documentary that explains today's topic in detail and that I recommend for you to watch. It's called Origin, and it's available in multiple languages. Here we see a model of a protein. And even though we see it as being solid and rounded, it's actually made up of smaller molecules called amino acids. There are 20 different amino acids, all with different sizes and different chemical characteristics. To form a protein, hundreds of amino acids join together in a specific order, forming a chain. When the chain is formed, it folds over itself, also adopting a specific shape that is related, this is related to the function of the protein. Okay. For the protein to fold and function properly, the amino acids have to be in a precise order. How is it possible to make such long proteins with only 20 types of amino acids? Well, because they're repeated. Amino acids are like the letters of the alphabet that are combined to form words or phrases. In Spanish, we have 27 letters, but with these letters, we can write very long texts. What is important for the text to make sense is the order that we put the letters in. Well, the same thing happens with amino acids and proteins. In this picture, you can see the 20 types of amino acids that form proteins. They differ in size and chemical structure, but they all have something in common. On the top part, you can see a small blue ball on one side and two small red balls on the other. For those of you who know some chemistry, you'll recognize it as an amino group and an acid group. And that's why they're called amino acids. This part that they have in common is what's used to form the chain. The acid group of one amino acid joins with the amino group of the next amino acid. Red with blue, and as it goes on. And the rest of each amino acid, the part that is different, is left hanging from the chain. And that's important because those parts can then interact with each other. Some amino acids have positive charges, and others have negative charges. Some are hydrophobic, which means that they don't like to be in contact with water, and others are the opposite. They're hydrophilic, which they love water. When a protein is made, the amino acid chain is automatically folded so that the amino acids are as comfortable as possible. The positives join with the negatives. The hydrophobic ones join each other and are placed in the center of the protein, surrounded by the hydrophilic ones, which protect them from the water, and so on. The final form of each protein depends on the type of amino acid it contains and, above all, on the order of those amino acids. If we change the order of the amino acids, that will change the interactions. It will change the folding, and it will change the final shape of the protein. Since the shape of the protein is related to their function, for a protein to work, it is very important that the amino acids are placed in the correct order. A protein that has the wrong or misplaced amino acids will not work properly. Do you remember when we talked about sickle cell anemia? Well, in people who are sick with this, the reason that the red blood cells are misshapen and can't carry oxygen properly is because the hemoglobin has one wrong amino acid. A cell contains thousands of proteins, all with super important functions. So the cell has to make sure that the proteins are made correctly. How does it do that? Well, it does it thanks to a cellular machine that is responsible for making proteins, and it's called a ribosome. Well, actually, it's about hundreds of these protein factories that are scattered throughout the cell. They're those little balls that you see everywhere in the picture, the ones that are being pointed out with the arrows. And if we go a little closer, we can see the ribosome more closely. And we see that it's made up of two pieces, one large one and one small one. Ribosomes make the proteins that the cell needs by taking amino acids and joining them one after the other in the correct order. But how do the ribosomes know what to make? How do they know which amino acids to use and in what order to place them? Ribosomes do their job very well because they do it by following precise instructions. Where do these instructions come from? Well, I'm sure that you've already guessed. It comes from DNA. DNA is often compared to the blueprint of an organism because it contains precise instructions for making every part of the organism. Specifically, DNA contains instructions for making proteins, each and every protein that the cell needs. But those instructions are encoded. We've already said several times that DNA is made up of 4 types of subunits, 5 nitrogenous bases, and we represent them with the letters A, T, G, and C. How is it possible that with only 4 letters, DNA can instruct ribosomes to build thousands of different proteins? It's easy to understand if we think about the binary code that our computers work with. All of the information that my laptop contains is encoded as a long sequence of ones and zeros. But if it's deciphered, I have videos, books, texts, music, games, and many other things. Humans have actually invented various systems to encode information using a small number of different symbols. Another very famous code is Morse code. It only has 2 symbols, dots and dashes, but it also allows us to encode and send any text such as this one, that you're going to hear right now. It says, "In the beginning, God created the heavens and the earth." If we wanted to, we could actually transmit the entire Bible in Morse code. And to decipher the message, we would only need to use this key that tells us how to convert the dots and dashes into the letters of the alphabet. The making of proteins from the information contained in DNA works in the same way. In Morse code, the dot and dash combinations correspond to certain letters. In DNA, the letter combinations correspond to specific amino acids. In this case, the letters are always grouped 3 by 3. For example, the combination of CCA codes for the amino acid proline, and the combination of AGT codes for the amino acid serine. Here you can see the key that we use to decipher DNA messages. It seems complicated, but it's actually not that complicated. It's used starting from the center and working outwards. For example, if we want to know what the letters A mean, we look for the A in the center circle, then the A in the next circle, and then for the A in the outer circle. And that brings us to the amino acid called lysine. Let's look at another example. What do the letters GTA mean? We start from the center, we look for the G, then the T, and then the A. And we arrive to the amino acid valine, or valine. That's not that difficult, right? We're going to do a little interactive exercise. I'm going to put up on the screen a 15-letter segment of DNA that codes for 5 amino acids, as well as the key to decipher it. When you know what the corresponding amino acid sequence is, you can write it in the chat. And when we have the first 5 answers, we'll stop the exercise and check to see if they're correct. Here it goes. Let's see who's the fastest. You're making your ribosomes. You have to put all 5 of them together, not just one at a time. Make sure that you write all 5 together in the same message. Very good. And it looks like we have the first correct answer. So I'm going to put the solution up here on the screen so that you can check and see if what you've written is correct. We know that we have at least one person who's done it correctly. The amino acids that are encoded in this short DNA fragment are in this order: tyrosine, alanine, glycine, serine, and arginine. This way you can check to see if they've been coded correctly. What you've just done is what ribosomes normally do, but they don't build chains of 5 amino acids, but much longer ones, of hundreds of amino acids, and they do it very quickly and without mistakes. In this picture, we can see the ribosome again, but now it's actually working. Between the two pieces, the small and the big piece, slides a kind of ribbon that carries the DNA information. The chain of balls that you see in the upper part is the protein that is being made following the instructions on the ribbon. If we could see the ribosome on the inside, we would realize how complicated the system is and how well it is made. The ribosome doesn't have to go looking for the amino acids to put in the chain because they are transported to it by transport molecules moving constantly through the cell. And they actually put them in the right places. And so that there are no errors, each of these transport molecules carries a single type of amino acid. And at the other end of the molecule, it has holes that fit with the 3 letters on the ribbon that correspond to the amino acid, as if it were a key in a lock. I'm going to show you an animation now of a working ribosome, so that you can actually see how quickly everything actually happens. Since everything is going to happen very quickly, I'm going to explain what you're going to see. Here you see the ribosome. The elongated yellow molecule that's in the lower part is the encoded genetic information. The green chain is the protein that is being formed, and the red things moving at full speed are the amino acid transporters. Let's see. Now we're looking from the outside, but we're looking on the inside after taking the top off, and we see what's going on the inside. We can see how the protein grows and the detail of how amino acids are added at full speed. If the genetic message and the letters are grouped 3 by 3, transporters bind to these groups of 3 letters in a specific way, and each one which it corresponds to, and releases its amino acid, which is added to the protein. Once the protein has been made, it is released from the ribosome and then automatically folds depending on the order of its amino acids. Isn't that amazing? Well, what you've just seen is a super simplified version of what really happens. The process of making proteins in the cell is much more complicated. In this drawing, we can see it in a more summarized way. The ribosome needs the information from DNA to make proteins. But DNA is so important to the cell that it is protected in a special compartment, the nucleus, that you see at the top left side, when it never leaves from that spot except for when the cell is about to divide. So when a protein needs to be made, what happens is that a copy is made of that part of the DNA that contains the information for that protein. And it's that copy that comes out of the nucleus and binds to the ribosome. The machine that makes copies of DNA is a protein, an enzyme called RNA polymerase. It's called this because the copies are the bits of DNA called messenger RNA. Amino acid transporter molecules are also copies of pieces of DNA and are also called RNAs. But since they transfer amino acids, they are called transfer RNAs. And if you look at the drawing, you can see that the ribosome, the protein factory, has 2 components. Proteins, of course, and another type of RNA called ribosomal RNA, which is also a copy of another piece of DNA. Last week, I told you that the DNA protein system was a magnificent example of interdependence. And now we just saw what I was referring to. And let's look what I meant. Our cells and our body are made of proteins. Proteins serve many vital functions. The functionality of the proteins depends on their shape. The shape of proteins depends on the sequence of amino acids. Proteins are made of ribosomes. Ribosomes are made of proteins and ribosomal RNA. Ribosomes arrange amino acids in order following the instructions of the messenger RNA and with the help of the transfer RNAs. RNAs are copies of DNA fragments. And the machines in charge of copying DNA and making RNAs are proteins. I know that this is pretty complicated, but we can summarize it in a simple way. DNA contains the instructions for making all cellular proteins. But to copy those instructions, read them, and translate them, proteins are needed. Let's imagine for a moment that chemical evolution was possible and that electrical storms and atmospheric gases had formed a few nitrogenous bases that by chance had joined together to form a piece of DNA. Let's continue imagining that by chance, the order of the letters was the correct one to make a small protein. And here is where we have to stop imagining, because if this were possible, which it's not, that protein would never be able to be made. Because converting a genetic message into a protein requires proteins that can copy DNA and make messenger RNA, and ribosomes that are also made of proteins. If we try to do it the other way around, and in our imaginary world a few amino acids have been formed first, which have given rise to a protein, the protein may last a while, but when it degrades, there will be no way to make another like it, because there is no DNA that encodes and stores that information. So without DNA, no proteins are made. But without proteins, DNA can't do anything. If what I have just told you in the last 5 minutes seems too difficult, don't worry. Genetics is a very complicated branch of biology, and there are many things that I don't even understand. But that's precisely the issue. An information coding system so sophisticated that even scientists have a hard time understanding how it works. How could it have all appeared all by itself? In fact, the very existence of coded information should lead us to the conclusion that DNA cannot be a product of natural and random processes. We know from experience that codes are invented by intelligent beings to efficiently store and transmit information. That's why binary code is used in computers. Morse code was named so because it was invented by a man named Samuel Morse with the intention of facilitating the transmission of information over long distances. And the genetic code is not just any code. Scientists have investigated it and found that it is an optimal code for several reasons. First, the groups of letters that code for each amino acid are not chosen randomly, but in such a way as to reduce the effects of mutations. If you look at the key that I showed you earlier, there are several combinations of letters that produce the same amino acid. For example, there are 4 possible combinations for proline. If when the DNA is copied, there is an error in the last letter, that mutation will not be a serious one because even if the letter changes, the amino acid will be the same. Even in cases where a letter change does change the amino acid, the code is made in such a way that for many mutations, The new amino acid is similar to the old one in its chemical characteristics. So that way the protein will then fold in a similar way and will probably continue to function even if it's not perfect. Second, and this is mind-boggling, scientists have discovered that the genetic code allows DNA to encode several different messages on the same fragment. Have you heard of palindromes? They're phrases that are read the same way both forwards and backwards. Here's an example on the screen in all the languages. And in English, it says, Eva, can I see bees in a cave? And if you read it backwards, it says the same thing. Well, there are pieces of DNA that can also be read backwards and forwards. But in this case, they contain different messages, which both make sense. If one side is read, it makes one kind of protein. And if the other side is read, a different kind of protein. And both are functional. In other cases, the same piece of DNA contains information to make several different proteins by taking only some pieces and leaving other pieces unread. Okay, I know that this sounds very complicated, but I have a fun example to explain it. In the days of when mobile phones didn't exist, people sent letters. This is a letter that fell out of the pocket of a Victorian gentleman and that someone decided to publish. It's addressed to a young lady, and because the quality isn't very good of the letter that you see here, I've transcribed it so that you can read it better. And I'm sorry that this only works for those of you who read English, but we're going to explain it for everyone else. The letter is quite unpleasant, actually. The gentleman tells the lady that he doesn't love her anymore, that he finds her boring, and to please not bother him again or try contacting him anymore. I'm going to leave you a few more seconds so that you can finish reading it. But it turns out that if we remove one line out of every two, the letter still makes sense, but it contains a completely different message. It's a hidden message, probably to be hidden from the young woman's parents. If we reread it like this, it turns out that he's actually declaring his undying love for her. Can you imagine how difficult it is to write a text like this? It takes a lot of intelligence to hide the love message inside the breakup message and for both of them to make sense. Well, we find the same thing in DNA. Many pieces of DNA contain several meaningful messages, one inside the other, and the cell is able to decipher all of them. These things, I get so excited talking about them that I could go on talking about them DNA for hours. But I told you that I would save time for questions today, so I'm going to start finishing up. I want to end with a quote from a very famous scientist, Francis Crick, who is one of the discoverers of the structure of DNA and who received a Nobel Prize just for that. Francis Crick says that biologists must constantly keep in mind that what they see was not designed, but rather evolved. Well, why would he say something like this? Well, because biologists, and especially geneticists, have to take or make a great effort to remain evolutionists. DNA, with all of its wonders, is like the signature or the seal of God in each of our cells. It's so obvious that ignoring it must be done consciously and intentionally. You have to repeat it to yourself over and over again. It's not design, it's not design, it is not design. Because if you are honest and study genetics with an open mind, you realize precisely the opposite. That what we biologists study is full of design, everywhere and at all levels. I think there is a verse in the Bible that's perfect to end with. It's in Psalms 139, verse 14, and it says the following: I will praise you, for I am fearfully and wonderfully made. Marvelous are your works, and that my soul knows very well. Next week, we will continue talking about the secrets of DNA with the topic epigenetics and pre-adaptation.